Hello everyone, welcome to the seminars in biotechnology. Today I would like to remind first we will start here in Zoom meeting for our guest presentation, and after that we will continue our seminars for student presentations in Microsoft Teams program. And today our guest is Professor Dr. Remziye Yılmaz from Hacettepe University Food Engineering Department. And briefly, I would like to introduce her. She received her PhD from the Hacettepe University in 2002, where her research focuses was molecular identification and characterization of lactic acid bacteria. And between 2002 to 2005, she founded and worked at the first molecular biology laboratory on the detection and quantification genetically modified crops at the Ankara Food Control Laboratory, Ministry of Agriculture. In 2005, she received a postdoctoral position from Middle East Technical University, Molecular Biology and Biotechnology R&D Center. From 2005 to 15, she conducted research as the head of the Genome Analysis Laboratory at the METO, where her research contributed to the understanding of the functional genomics and genetically modified organisms, including microorganisms. And in 2006, short term, she moved to Wageningen University and Research, where she was a Professor Dr. Leo de Graaf postdoctoral fellow in the Laboratory of Fungal Genomics. During this time, she expanded her research interest to focus on the functional genomics of plants such as wheat and barley using the microarray analysis model. And today she will present with the title of Risk Assessment of Genetically Modified Plants and Derived Food and Feed. So, Remziye Hocam, speech is yours. I will leave it to you.
Thank you so much. Thank you so much for your invitation, kind invitation first. And I'm so happy to be here right now. And my topic is Guideline for the Risk Assessment of Genetically Engineering Crops and Derived Food and Feed. And I write here my name, my university, but Banu Hoca said that I'm originally coming from food engineering department. Also, I am coming from International Food Biosafety and Biotechnology Center. It is very important center for the Hacettepe University. It's established under the Hacettepe University in 2017, and it is a multidisciplinary center that shares knowledge about the global food sustainability and development. I would like to say something about our little bit new center. And this is my outline, my presentation outline. First, I would like to talk about what is risk and risk assessment, and after that, the need for a technical guideline and preparation process a little bit, and what is the content of the technical guideline, and after that, final remarks. And if you are a scientist or you want to be a scientist, you know very well what is risk and risk assessment actually. But generally, risk is defined as the likelihood effects will result from exposure to a substance, and risk assessment can be defined as a process of evaluation, including the identification of the attendant uncertainties of the likelihood and severity of an adverse effects occurring to man or the environment following exposure under defined conditions to a risk resource. And you can see here this picture, risk assessment graphic. It is including hazard identification, hazard characterization, and exposure assessment. This is the essential. And after that, all of them, risk characterization is the is becoming here. There are general principles for the risk assessment, and risk assessment should be carried out in a scientifically sound and transparent manner, and can take into account expert advice and guidelines developed by relevant international organizations. This is very important thing. First thing for the risk assessment, and also everyone can talk about the lack of scientific knowledge or scientific consensus about the risk assessment. This is the important, and the risk especially associations with genetically modified organisms or products, and thereof should be considered in the context of the risk posed by the non-modified recipients or parental organisms in the likely potential receiving environment. And lastly, risk assessment should be carried out on a case-by-case basis, because we need information may vary in nature and level of detail from the case to case, depending on the GMO concerned, and this is important for the risk assessment. Okay, why we need for a technical guideline for the risk assessment of genetically modified organisms? Because we have national biosafety systems, and these national biosafety systems vary from country to country, but it is usually have a number of common components, such as a policy on biotechnology, including biosafety, and the regulatory framework for biosafety, and the implementing and facilitating mechanisms. And this facilitating mechanism including system to handle notifications, informants and monitoring, public information and public participation, it is important, and mechanism to review. There are some questions about new technology you have to answer. First, is it useful or is it safe? These two questions are so important. Also, government policies aim to maximize benefits and minimize risks. That's why risk analysis is including risk management, risk communication, and risk assessment. I said before, risk assessment related to science, because you have to do risk characterization. That's why this topic is related to science, but risk management is absolutely related to policy, and the risk communication is absolutely related to information exchange. And you can see here risk assessment, risk management, and risk communication topics a little bit more. And I told you before about the science-based risk assessments subtopics, and here you can see here risk management subtopics. One of them is evaluation and the management option assessment, implementation of management decision, monitoring and review, and risk communication. I think this is very important topic for the Turkey, our country, and this is the actually interactive exchange of information and options on risk among risk assessors, risk managers, and stakeholders. We prepared a technical guideline for the risk assessment of GMOs in Turkey in 2017 again. And before preparation of this guideline, 2 workshops organized at the international level and national level in Turkey. And the guideline prepared under the roof of the support of implementation of the National Biosafety Framework project, and does not have any regulatory status, but elaborates on the information needed for the risk assessment of the GMOs, and just it seeks to provide guidance to both notifiers and risk assessor, and also aims to assist notifiers in the preparation of dossiers. And that's why it is important for us, and the risk assessor or the regulator may require additional information on a case-by-case basis, and this is also important thing for us. And notifiers must adhere to the requirements laid down in the appropriate biosafety law and related regulation in Turkey. Technical guideline for the risk assessment of GMOs published in 2017, and regulatory, it is including some topics such as regulatory systems for GMOs, technical guideline for the risk assessments, including Cartagena Biosafety Protocol, National Biosafety Law, and main elements for the risk assessment procedures for the regulated products in the concept of National Biosafety Law. And also we put a case study of safety assessment about the GM corn event such as NK603 MAIZE. And this is important part to understand what is the case-by-case evaluation. And also we put a glossary at the end of this book. What is the regulation process in Turkey? Actually, they started 1998 first with the acceptance of instructions of field trials for transgenic crops. After 1 year, the first field trials of potato, corn, and cotton varieties you can see in Turkey. And after 1 year from this, risk assessment process is implemented to evaluate safety of these GM varieties. And from 2002 to 2005, the implementation of the National Biosafety Framework project conducted as a part of the Global Environment Facility, GEF of UNEP, and this is important issue for Turkey. In 2010, biosafety law prepared by a commission composed of 27
Birisinin mikrofonu açık sanıyorum. Hocayı duyamıyoruz.
In 2010, biosafety law prepared by a commission composed of 27 experts from different ministries, universities, and non-profit organizations. And this law puts main elements of the risk assessment for all applications for placing on the market of GM organisms, and whether they concern the organisms themselves or food and feed products derived from them. The important law for the biosafety, and this biosafety law including some articles, and Article 2 is related to risk assessment actually, and it is including first some definition and the scientific methods items here in at the Article 2, and specification, evaluation, and determining of the risk factors, risk resources, and risks that may be resulting from the GMO and GMO products on human, animal, and plant health. And this is the important. And Article 3 in this law there is a summary of evaluation process for approval of GM organisms in Turkey, and there are some differences between nowadays procedure right now, and you can see here submission application first, after that the name of the ministry is changed right now. There is no food anymore, and Ministry of Agriculture and Forestry, and the biosafety board is cancelled right now, and the dossier is going to directly scientific committee, and after the 9 months for risk assessment, public consultation, and final decision from the TAGEM. TAGEM is the general directorate under the Ministry of Agriculture and Forestry, and they are responsible for the policy, agricultural policy and research mainly, and this TAGEM is the authorized under the Ministry of Agriculture and Forestry the final decision. And the public consultation is about 21 days. Article 4, risk assessment. In this law, risk assessment, very important topic, and they divided 2 different topics under the Article 4, because the risk assessment, they are thinking about the risk assessment is including scientific risk assessment and the social economic risk assessment. That's why you can see 2 terms here, but they are insist the scientific principles and they are made separately for each applications, and in applications different risk assessments are made separately for each application, and risk management principles are designated based on risk assessment and the social economic risk assessment results for applications of GMOs and GMO products. This is the essential again. And there are some prohibitions in this biosafety law at Article 5. And you can see here, making production of genetically modified plants and animals is prohibited, and the placing GMO and GMO products on the market without approval also prohibited. And there are 5 prohibitions. They are so important about the baby food and the commission scope and place on the market, and violation of commission decision. This is very important part of this law. And you can see here in this slide main elements of the risk assessment procedures for regulated products in the concept of national biosafety law. And one of them is dossiers to be submitted, and the secondly risk assessment, and lastly the final report. The final report including structure, including some information about the GM structure, GM molecular characterization, food and feed safety, and environmental and conditions, management. Would you please close your...
Arada birkaç kişinin evet mikrofonu açıldı. Remziye Hoca zor durumda kalıyor. Ayyüce mikrofonunu kapatır mısın?
Thank you so much.
Pardon hocam.
No problem. Okay, but what is the current situation in Turkey? Turkey make use of the international organization standards such as OECD or FAO in developing scientific risk assessment of biosafety and in shaping its national regulatory system. And it has technical expert body to conduct risk assessment of a product seeking regulatory approval. For the GMO analysis, each laboratory should be established the verification on method performance criteria and calculation of measurement uncertainty. This is the essential for the monitoring of the GMOs. And also, as in the EU, food and feeds containing greater than 0.9% GM organisms or their products are labeled in Turkey. And today, Turkey has approved 13 soybean, 23 corn, and 3 microorganisms as GMOs currently for feed use only. And they are using these 3 microorganisms for the enzyme production. And they got approval 1 year ago maybe, and they are producing enzyme for food also. This is a little bit different from the feed. And I would like to show you a case study to understand about what is the risk assessment. This is the safety assessment of NK603 MAIZE event. First, I said before you, first you have to define the NK603 MAIZE transformation material. What is the gene encodes? What is the gene? What is the how can you do transformation for this MAIZE? And what is the function for the EPSPS gene here? And what is the biosynthesis pathway? And this is very important for each case and also NK603 MAIZE here. And gene cassettes also so important because you can use the CP4 EPSPS gene, but you have to use some promoter and terminator genes for the gene cassettes, and they have to know, risk assessor, they have to know what is the promoter, your promoters and terminators, and how many copies are there in this event, and what is the form, product, and the function. What is the function with these gene cassettes? That's why notifier applicants has to give this information for the risk assessment. You can see in this slide some methods for the molecular characterization, and sequencing inserted DNA is essential for this event. I said to you before, 2 intact CP4 EPSPS gene cassettes within the single insert. You have to show these results with the sequencing methods about the inserted DNA, and also PCR and DNA sequencing are essential. Western blot and RT-PCR are the essential for the molecular characterization. Inheritance study and stability tests are very important topics for the molecular characterization study of the GM events, because there are so many discussions about the inheritance and the stability of the GMOs in the world right now, and also in Turkey, and that's why stability tests is very important topic for the risk assessment. Also, you have to see some ELISA tests about the CP4 EPSPS protein levels, and you have to assess these results during the risk assessment. And this slide is related to safety assessment. This is a little bit summary. And NK603 MAIZE event, these all information are related to only one event, because we are doing the risk assessment case by case. This is only related to one event. And we are starting the protein characterization and the safe long history. We are wondering about the long history of safe consumption of similar proteins, digestibility, in vitro digestibility, and lack of toxicity, acute oral toxicity in mice, about the specific protein with this specific MAIZE event. And you can see here several methods, and they have to show us their results, and we have to discuss very long time on this dossier related to results. Okay, this is the going on the compositional analysis and nutritional assessment. This is very important for the food part and feeding part. And you can see here compositional analysis, the including some subtopic, and nutritional assessment also. Here, the feeding animals are so important for the nutritional assessment, and there are still some discussion about the feeding trial studies right now, but you have to see, you have to evaluate these nutritional assessment results with the any application also, and for the NK603 MAIZE event. And this is the general approaches for the risk assessment, case by case risk assessment, but you can see here a summary of final reports written by Scientific Committee of Turkey for NK603 MAIZE. And this report is content is including first vector construct and transformation process. It is including analysis of molecular structure, expression, and stability of inserted gene. And secondly, risk analysis of chemical composition and agricultural properties. This is very important for the Turkish Scientific Committee also. And toxicity assessment, allergenicity assessment, environmental risk assessment, and also food processing technologies. And lastly, risk management. I said before you, risk management part is essential because of the biosafety law. And you can see here this summary and the final decision. You can see here two parts for the final decision, because during the biosafety board decision was the final, but today this is different, because the TAGEM is the essential, Ministry of Agriculture and Forestry essential for the final decision. And the scientific committee decision is, it is concluded that the use of corn for the production of fully refined oil, sugar, syrup, dextrins, and starches may not pose risk when GM NK603 corn and its products are used as safe as food. And biosafety board decision, but this is different right now, I said before, application of NK603 corn for food use was withdrawn by applicants. Therefore, there is no final decision of biosafety board on this event. And the final remarks for my presentation, and the technical guideline. I think the technical guideline for the risk assessment of genetically modified plants and derived food and feed. It is important for the Turkey and also they can use risk as a risk assessor and notifiers who intend to apply for the commercial release of genetically modified plants in Turkey. And this is important for the commercialization procedure of the GMOs in Turkey, this document does not cover genetically modified animals or microorganisms or medicinal products for human or animal use, and this is important because there are some more need for the new technical guideline on these issues right now in Turkey, and at the same time issues such as containment or risk management are not within the scope of this document, and thus the post-market monitoring of GM crops and derived food and feed, it is not addressed specifically. My last word about this biosafety regulations, I write this paper before about the Aesop's famous fable. You know very well tortoise and hare, Aesop's famous fable, I think. And it taught us tortoise wins by taking slow, steady approach against its fast, overconfident competitor hare. I propose tortoise strategy is more beneficial when it comes to public perception and zero tolerance toward modern biotechnology research and development for sustainable food and agriculture in developing countries. This is my last word. And I want to say again, I would like to thank for First Assistant Professor Dr. Ceren Bayraç, who is my co-author for this guideline, and our advisor, Professor Dr. Rebecca Grumet from Michigan State University, and also Biotechnology Group Institute at the Gebze Technical University. And thank you so much for your kind invitation and kind attention here. Is there any question from you? I'm ready. Thank you.
Thank you, Remzi Hocam, for this nice presentation.